Friends, welcome to Biology Made Easy. Today in this video, we will discuss codominance. So what is codominance? The two alleles of a gene, when they express themselves simultaneously in heterozygote, that is known as codominance. That means usually we have seen that when a condition is in heterozygous condition, according to Mendel's law of dominance and recessiveness, the capital T is dominant over small t, so it will express as tall. In incomplete dominance, what we have observed in the one of the videos, in that case, both capital T and small t, both of them are unable to express themselves. This heterozygous condition, it is neither tall nor dwarf, and it gave an intermediate character. But here in case of codominance, both of the alleles, when they are present in heterozygous condition, both of them will express simultaneously. That is the difference between incomplete dominance and codominance. Usually, the codominance differs from incomplete dominance in what respect? One is each allele produces its own effect. That is number one. It differs from incomplete dominance. The second one, the two phenotypes remain side by side. And the third difference from incomplete dominance is they do not blend with each other. They do not blend with each other. They do not mix with each other. So, in these three aspects, the codominance differs from incomplete dominance. That means they express themselves independently. So, let us take some examples. Now, the example number one we can take is the AB blood group in case of human beings. Example number one, the AB blood group in human beings. So, all of us know that for the blood group, usually for blood group A, usually the gene is IA and it produces antigen A. If the blood is B, then it's IB and it produces antigen B. Now, in case of blood group AB, it is IA and IB. That means here AB are superscript. Then what is the antigen? Antigen both A and B. That means here both IA and IB, they are expressing themselves simultaneously in the AB blood group. Now it will make a cross between a homozygous A blood group with a homozygous B blood group. Then it will be IA, and it will be IB and IB. Now, when two such persons marry with each other, then the offspring will be IA, IB. So, now it has blood group. The blood group is AB. That means both A and B express themselves. Now, in the next generation, what will happen? F2 generation. In the F2 generation, IA, IB, and this side also IA, IB. One side male or the other side is the female. So it will be IA, IA. It will be IA, IB. This will be IA, IB, and this will be IB, IB. That means in this case, you see, what is the blood group? Blood group A is to blood group AB is to blood group B. They are in the ratio of 1:2:1, because this will be A blood group, this will be B blood group, and this will be AB blood group, this will be AB blood group. That means A, AB and B, they are in the ratio of 1:2:1. This is the phenotypic ratio. And if we will come to the genotypic ratio, the genotypic ratio genotypic ratio we can write like this: IA, IB, and IB. Genotypic ratio. So this is also again in the ratio of 1:2:1. That means both phenotypic and genotypic ratio are in the same ratio 1:2:1. Now this same case is also found in case of sickle cell anemia. Now I will take the example number 2. So example number 2 is sickle cell anemia. So in case of sickle cell anemia, if we make a cross between or even two persons, one is a normal person will marry with a sickle cell person, then what will happen? Then the offspring will be HbA and HbS. That means a carrier. That means this person is neither normal nor sufferer of sickle cell anemia, is carrier. Now, if two such persons will marry, then in the two generation what will happen? HbA, HbS. This side also HbA and HbS. Now see, this one HbA, HbA. This will be HbA, HbS. This one will be HbA, HbS. And this will be HbS and HbS. That means this person is normal. This person is a sufferer from sickle cell anemia, while these two are carriers. That means here, again they are coming in the 1:2:1. That means the normal 1, the carriers 2, and the sufferer 1. This is the phenotypic ratio. Again the genotypic ratio. The genotypic ratio HbA 1, HbA HbS 2, and HbS 1. That means both phenotypic and genotypic ratio are same 1:2:1. Now let us give one third example from that of animals. In one breed of cattle, we will find one interesting example, that is the coat color in one breed of cattle. So example number 3, coat color in one breed of cattle. So here one breed is a pure black, or we can write the black cattle, we can write black cattle represented by capital B, and crossed with white cattle written as small b. Usually what happens in the black cattle, we will find there are the hairs which are black. Black hairs are there. And in case of white cattle, there are hairs, but these hairs are usually white. Though I am drawing green, but they are white. Now when the cross is made between two such cattle, then the F1 hybrid will be capital B, small b. So if it would have been Mendelian cross, it should have been black because capital B would have been dominant over. But here in case of codominance, this is Roan color. Roan color. That means a different color. Now when two such, and in this again what happens? What will be the hairs? The hairs will be intermixed. Black, 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 and you will get white hairs. That is, black and white hairs are intermixed. That is the Roan. Intermixed black and white hairs. Now, when two such are crushed, intercrusted in the F2 generation, what will happen? When these two will be intercrusted, then in the male side will get capital B small b, in the female side also will get capital B small b, and now this will be capital B, this will be capital B small b, this will be capital B small b, this will be small b small b. So when this one is what? This one is black. Now what about these two? These two are Roan. What about this? This one is white. That means black one, Roan 2, white 1. That means phenotypical the ratio is 1:2:1. Now come to the genotype. The genotype is capital B, small b, and small b, small b. So you say this is 1, these are 2, and this is also 1. That means both phenotypic and genotype ratio are same. So these are the different examples of the codominance where both the alleles express themselves simultaneously. They do not blend together, nor there is a dominant and a recessive character like that of Mendelian experiments. Now let us discuss questions. So I can write here the MCQ. So question number 1, which Mendelian idea is depicted by a cross in which F2 generation resembles both parents. So this question was asked in NEET 2013. So what are the choices? First choice is incomplete dominance. Second choice is codominance. Third choice is law of dominance. And the fourth choice is inheritance of one gene. This is one question. Now come to the second question. Codominance alters the expected monohybrid. First choice F2 genotypic ratio. Second choice F2 phenotypic ratio. Third choice both A and B. And fourth choice test cross ratio. And this question also, it was asked in AIPMT. Now let me give a third question. Third question is codominance. Codominance differs from incomplete dominance. Incomplete dominance in first choice F2 phenotypic ratio, second choice F2 genotypic ratio, third choice not being blending inheritance, and the next one is being blending inheritance. This question also asked in AIPMT. So these such type of questions are usually asked in all types of national examinations. I hope now you have a very clear understanding about codominance. Thank you very much for watching the video. God bless you. Have a good day.